The next lesson, lesson 6, is about reproduction and genetics. The first thing is cell division. Cells have to reproduce one after another. If you have one skin cell, well then it reproduces into many skin cells. That way when skin cells die, the new ones form. They say that you get a brand new body once every 6 weeks or so because all the cells in your body at one point or another die and then are recovered, but replaced by other cells. And the process by which this is done, the process by which normal cellular reproduction happens, is called mitosis. M-I-T-O-S-I-S. And mitosis works very simply. You've got an original cell over here. You can see these things in here are the chromosomes. A human being actually has 46 chromosomes. This diagram only has 2, but that's okay. You can imagine it. And what happens in the process of mitosis is the cell it doubles itself. Then the cell itself splits apart, and you end up with 2 daughter cells that are the same as the parent cell. 2 diploid cells, which means double, that have the same number of chromosomes as the original. In terms of organizational reproduction, in other words, the entire organism reproducing, you have sexual and asexual reproduction. Sexual reproduction simply means you have 2 cells, one from 2 different organisms, one from a male cell and one from a female egg cell, and they combine to form an offspring. Asexual reproduction is simply where you have one offspring that gives rise to another one. Let's look at them one at a time. Mitosis, which we just looked at, this is the regular cell division that happens in your body, and the way that small cells like bacteria and things like that split apart into 2 daughter cells. Diploid cells, which means again cells that have the same number of chromosomes as the parent cell, have a pair of homologous chromosomes, which means chromosomes that go together with each other. I mentioned before that human beings have 46 chromosomes, and that's because we have 23 pairs of chromosomes. 23 they're not twins, they're different from each other, but 23 pairs of chromosomes. Meiosis is how we produce our reproductive cells. Our reproductive cells only have half the number of chromosomes as the parent cell. So for example, in an organism like a human being or a mammal or even most trees, you have a male reproductive cell known as a sperm cell and a female reproductive cell known as an egg cell, and each of them have half the number of chromosomes. So for example, in a human being, a sperm cell and an egg cell will each have 23 chromosomes, whereas a full organism has, of course, 46 chromosomes. Here's a little diagram like the one that we just saw. We've got mitosis on the left side, which is a parent cell. It replicates and it comes out to 2 equal daughter cells. Whereas over here you have meiosis, where a reproductive cell is being formed, and notice that the daughter cells only have half the amount of chromosomes as the parent cell, and eventually you have a process of fertilization where the 2 cells join, and this over here is what is called a zygote, which means the first cell in a brand new child. Here's a little bit of a bigger diagram over here of meiosis, where you have a parent cell, DNA replicates into 2 daughter cells, and each process produces 4 cells, 4 reproductive cells that can then be used to reproduce eventually. Now, of course, there are some problems that exist when things go wrong. One very famous example, is the example of Down syndrome. Down syndrome is, as we discussed before, there are 23 pairs of chromosomes, and Down syndrome occurs when through some sort of a problem with the reproductive process in meiosis, an extra copy of chromosome 21 is there, and it affects about 1 in every 700 people, and people who are affected by Down syndrome very often have mental retardation problems and shortened lifespans. This happens when one pair of chromosomes fails to separate during meiosis. Remember, during the meiosis process at the beginning over here, really in this second stage over here, the chromosomes split into half the number as the parent already had. Now, if one of them fails to split, so instead of the daughter cells, let's say, having 23 chromosomes each, they each have 24 chromosomes, then you're going to have too many chromosomes in the child. Instead of having 46 chromosomes, the child's going to have 47. And the problem with the 21st chromosome not splitting is something that specifically leads to Down syndrome. Here's really the example. You have normal meiosis, this is where they split apart to have half the number of chromosomes, as you can see over here. If I know it's very small, I'm sorry for that. But what happens in the right side, in the Down syndrome case, is 24 chromosomes split normally at the beginning over here, but look what happens to the second stage. Instead of being only on this side would be the normal area, one black and one orange on both sides. Over here you've got two blacks and one orange, and this cell eventually goes to fertilize and become a child, and that child could have Down syndrome. Now we're going to move to genetics. Genetics is the study of how characteristics are passed from one generation to another. You probably have noticed that parents who are tall tend to have children who are tall. Parents who are smart tend to have children who are smart. Parents who have darker skin tend to have children who are darker skin. It's because of the idea that parents pass their DNA. The parent's DNA goes to the offspring. About half of the DNA comes from the father and half of the DNA comes from the mother. And the reason is because diploid organisms, which means organisms that have two pairs or pairs of chromosomes, have pairs of, well, pairs of chromosomes. Homologous means they form pairs. So we have 23 pairs of chromosomes. Each of our 23 pairs, one of the chromosomes came from our father, and one of the other chromosome came from our mother. So they have two copies of the same gene. If both have, now an allele means something that expresses a particular trait. Let's take, eye color. Eye color, there are three basic eye colors. There's blue, there's green, and there's brown. Hazel is a combination of green and brown, but those are the three basic, most common eye colors. Now, an allele would be, let's say, for example, a gene for a green eye. So if you have two genes for green eyes, your mother gave you a green eye gene and your father gave you a green eye gene, then the organism is what is called homozygous. Homo meaning the same. It's the same. They both alleles, both genes are the same. Both are for green eyes. If the homologs have different alleles, the organism is heterozygous, which means it's mixed. You might get one green eye gene from your mother and a blue eye gene from your father. Now there is a distinction. You have one green and one blue. Now we'll discuss in a minute which one you actually get, but it's heterozygous because you don't have the same one from both parents. A genotype means what the gene actually is. If you have a green eye gene and a blue eye gene, then your genotype is one green and one blue. Which one is actually expressed is called the phenotype. For example, let's say a cell in a person's eye has the genotype for brown here, blue eyes, not have hemophilia, and a lot more information. In this example, this is really based on this similar idea that every cell in your body has the DNA to express every gene in the entire body, but only some of them actually are expressed. The gene in your eye has DNA that tells your feet how to behave too, but only expresses it based on only is relevant to your eye. I'm sorry, I skipped a couple of slides over here. We'll just go over this. I mentioned this already, but let's mention it again. Genes, a gene is a segment of DNA that are the units of inheritance. DNA is made from proteins and produces proteins, cells, and really the entire organism. Every cell in your body has the DNA really to express everything in the entire body. Locus is the gene's location on the chromosome. At a particular point on the chromosome, you have the gene for your eye color. At a different point in the chromosome, you have a gene for your hair color. At a different point, you have a gene that illustrates your height. And an allele could mean different versions of the gene at a given point. A trait or an allele could be dominant or could be recessive. Dominant means it shows up, and recessive means it doesn't show up. Just to give you one example, blue eyes are recessive as to brown eyes, which are dominant. If a person has 2 brown eye genes, they're going to have brown eyes. If a person has 2 blue eye genes, they're going to have blue eyes. But if a person has one brown and 1 blue, they are going to have brown eyes because brown eyes are dominant and blue eyes are recessive. Same thing is true with green and blue. Green, if a person has a green and a green, they're going to be green eyes. A blue and a blue is going to be blue eyes. But a green and a blue, they're going to have green eyes because green is dominant over blue. Green and brown are actually codominant. That's why if somebody has a green and a brown eye gene, they're going to have hazel eyes, which is kind of a combination of green and brown. This is the way, basically, a pair of chromosomes look. You got 2 chromosomes that are a homologous pair, and on each point on the gene over here, you could have 2 alleles that go for the same thing. Like this green area and this green area both establish hair color. They could be the same, in which case you have the homozygous trait, or they could be a heterozygous trait. And again, as we discussed before, the genotype is what you actually have in terms of the gene. The phenotype is how the gene is expressed physically. You can also use what are called Punnett squares to make a determination of what the odds are of a particular child of particular parents having a particular gene. This is based on the idea of what is likely, what are the odds of a particular trait being passed to children. Basically, I mean, I'll give you examples over here, but the easy way to draw the Punnett squares is to draw a box with 4 squares, and for each possibility, you draw them on top and on the side. Well, I'll put that as an example over here. Without an example, it's impossible to figure out what that means. Let's take an example. A father has 1 dominant and 1 recessive gene for brown eyes, and the mother has 1 dominant and 1 recessive gene for brown eyes. We'll call big B brown and little b blue. What is likely for the child to have? Now, for this Punnett square, you put 1 parent up here and 1 parent on the side, and the father has 1 big B for brown and 1 little b for blue. That's the father's genotype. And the mother's genotype is also 1 big B and 1 little b. So you kind of draw a little bit of a grid over here and see what the combinations establish. This box is a combination between this and this, which is 2 big B's. This box is a combination between this and this, which is 1 big B and 1 little b. This box is a combination of this and this, which is 1 big B, 1 little b. And this box is a combination of this and this, which is 2 little b's. So we see over here there's 1 homozygous brown, 1 homozygous blue, and 2 mixtures, 2 heterozygous genes. Now the 2 heterozygous genes are both going to end up with brown eyes because brown is dominant. So in this example, if you have a father that's heterozygous brown and a mother heterozygous brown, the odds are going to be about 75, since either the chances are equal that the father will either give a big B or a little b, and the chances are equal that the mother will either give a big B or a little b. The odds are 75%, namely 3 out of 4, that the child will have brown eyes, and 1 out of 4, namely this guy in the corner over here, that the child will have blue eyes. If we had, other examples, let's say, for argument's sake, you're talking about, let's say, again, let's try capital B for brown. Let's take this example where a capital B is brown, a G is green, and a B is blue. Remember, B and G are both dominant because they give you hazel, but either one of them is dominant over blue, which is recessive. So let's say, for example, you've got a father who is brown heterozygous and a mother who is green homozygous, in other words, 2 greens. Now, if you wanted to know what the child would be, then you would draw a little Punnett square over here, and you would take the 4 combinations. In this box, you've got a B and a G. So this is, well, let's do all of them. This one is G and B. This one is also BG, and this one is also GB. So these two would both be brown and green, which are codominant, and you'd have hazel. That's 50%. Half of them are hazel. And this half would be green and blue, which turns out to be green. Also half of the children. Let's take a different example. What if instead of, I'll tell you what I think just my own family, I believe, is that, I mean, I have particular reasons to believe this, but I think my father happens to be heterozygous brown, so a brown and a blue. My mother, I believe, is heterozygous green, which is a green and a blue, which means in my family, what do we got? We got one chance for BG, which is hazel. That's what I have. We've got a GB over here, which is green. My sister has that. We've got a BB, capital B, lower b, which is brown and blue, which is brown. That's my brother. And the last one is BB, blue, blue, which is possible for a blue-eyed, and that's none of my siblings. I've got two siblings, so we've got this one, we've got this one, we've got this one, but we've got no blue-eyed. But again, these are just odds. It doesn't mean that if there was one more child in the family, the child would definitely be blue eyes. These are just based on the odds. And in case you're curious, the reason why I'm pretty sure that my sister is green and blue is that when she was a kid, she always had blue eyes, and they turned greenish as she became older. I think that what that means usually is that you have a blue-eye gene, and it takes the green-eye gene a while for its dominance to assert. It takes a few years for its dominance to assert. Family charts and things like that, sometimes known as pedigrees, can actually help you see which types of genes, specifically diseases, very often used for, are carried in a particular family. Now this chart happens to be, it looks like a royal family, looks like a German and British royal family over here that happen to have a lot of hemophilia in the family. Hemophilia is a disease that the blood doesn't clot properly, and it could be very dangerous if you get a cut, get a deep cut, something like that. You could actually bleed out from it. And this actually, the disease happens to be on the X chromosome. And males, we didn't discuss this before, but a male has a X chromosome and a Y chromosome, whereas a female has two X chromosomes. And the disease is recessive, which means if you have one good X chromosome and one bad X chromosome, you do not have the disease, which means it's a lot easier for men to get the disease than women, because women have two shots at a good X chromosome, and men only have one shot at a good X chromosome. If a woman has one X chromosome with the disease and one without it, the woman is called a carrier, because she has the X chromosome for the disease, but she doesn't actually show the disease. She's not actually sick. Again, just the point is, this is an interesting chart where it shows the clear boxes are men who do not have hemophilia. The black boxes show men who do have hemophilia. The white circles have women who don't have the gene, and the half-shaded circles show the women who are carriers, who have one X chromosome with hemophilia and one without hemophilia. This is one example over here. So scientists can figure out who the carriers for a disease are, and who wasn't a carrier, and who had the disease. Sometimes, there's an organization which many of you are probably familiar with called Darya, and what they do is they try to see who's a carrier for which disease. And if there are bad diseases like Tay-Sachs, familial dysautonomia, and certain other diseases that they test for, if two parents, if only one of them is a carrier for the disease, it's not a problem, because the child can then at worst only be a carrier. Let me draw that example. Let's say you have, let's say an H for healthy, and a C for a carrier. So if you've got two parents and one of them is healthy totally, and the other is a carrier, neither of them are going to have the disease, because the disease is only recessive. it's not dominant, so you won't see it. Then if you notice, the 4 possibilities for the children are healthy, healthy, and over here is also healthy, healthy. Over here is healthy and carrier, and over here is also healthy and carrier, but none of the kids are sick. At worst, the kids are carriers. On the other hand, if you have 2 parents who are both carriers, in other words, you've got an H and a C, together with an H and a C, well, 3 out of the 4 kids are going to be okay. You're going to have an HH, which is not even a carrier. Over here, you're going to have an HC, which is a carrier. Over here, you're going to have an HC, which is a carrier. And over here, on the other hand, you're going to have double Cs, which means you're going to have 2 carrier genes, which means the child is going to be sick. Which, if there's a disease like this, like Tay-Sachs, which is a recessive disease, but you have 2 carriers, there's actually a 1 in 4 chance that each child will have the disease. And that's what an organization like Dar Yesharim does. They'll tell the parents that this shidduch is not really a match, because the children could end up having these very difficult recessive diseases. This example over here and previously, this example is different than the one I just gave. The one I just gave is where the disease is on just a random gene out there, whereas over here you're talking about a disease that is on the X chromosome. You have the over here, Louis XIV was not a carrier for disease. Where's Louis XIV? I can't even find him. Somewhere over here. Here we go. Louis XIV, right over here. Then we had Alice, Prince of Hesse, or Hesse, was a carrier of the disease, right over here. I guess she married Louis XIV. And their children, you can see, the children either were carriers, totally healthy, or poor Frederick over here actually had the disease, because he must have gotten the X chromosome. The male cannot get an X chromosome from the father, can only get a Y chromosome from the father. Essentially, let me take a few moments to discuss that as well. The way gender determination works is that men have an X chromosome and a Y chromosome, and a woman has 2 X chromosomes. So when there's fertilization, the male always has 1 X and 1 Y, whereas the female has 2 X's. And you can see the odds if you draw a little Punnett square over here. You've got X and X over here. You've got XY over here, XX over here, and XY over here. So, of course, half of them are men and half of them are women. That's why, of course, the odds of a baby being a boy or a girl are roughly equivalent to each other. Now, if there is a disease on the X chromosome, a boy can only get an X chromosome from the mother. Because if it's a boy, it means they're getting a Y chromosome from the father by definition, which means it must get its X chromosome from the mother. So if the mother is a carrier, like Alice over here, she's got 2 X chromosomes, one good one and one with hemophilia on it. So Frederick got the Y gene from Louis XIV, but he got the X gene from Alice, which means he had a 50-50 chance of being sick, which means he was okay, but his brother Ernest, he was not okay. He was a hemophiliac, but his brother Ernest was okay. Okay, self-fertilization, there are some plants that actually have the male and female, even some animals, believe it or not, like sponges and things like that. Well, not sponges, but certain other very, very basic types of animals that have both the male and female parts at the same time. And trees very often do that, and different types of plants as well. And an organism's sperm fertilizes its own egg and does sexual reproduction, even though it's only one organism. The person that originally did all of these experiments to determine these Punnett squares that we discussed was an Austrian monk named Gregor Mendel. You might remember him from your high school biology class. And he did all these tests. We don't have to go through them in exhaustive detail, but he did all these tests to determine the effect of recessive and dominant genes. Okay, so here are some examples. We did some examples before. I don't think we need to do any more of these examples. Cell reproduction could also go wrong. It's called when cell reproduction gets out of hand and reproduces beyond control and just goes crazy and grows too big and interferes with other cells, that is what is known, unfortunately, as cancer. Mutations, which means some sort of a problem in the mitosis process, in the process of the cells reproducing. Mutations happen all the time. One individual mutation is not a problem. Usually, if there's a mutation, some sort of a problem, and it causes a problem with the cell, then the body just recognizes it, crushes it, destroys it, and flushes it out of the body, and that's that. You might have 100 mutations a minute. It's not a big deal because the cell can recognize them, crush the cell. It's only one of many, many billions and trillions and who knows what's number of cells you have in your body. So one cell is no big deal. But if it causes uncontrolled cell divisions, you have a lot of cancerous cells, or the cancerous cells start to get out of hand, obviously that could cause a big problem. One of the ways they try to treat cancer is to focus in, either with chemotherapy or with radiation therapy, to try to kill the cancerous cells. Now, unfortunately, the problem with both of those approaches is that they may kill the cancer cells, but first of all, they might not be able to kill every single one of them, and so it might come back later on. And second of all, those sorts of therapies also attack and kill the healthy cells that are nearby, which causes all sorts of problems to the patient. And let's take a look at some multiple choices quickly over here. Which of the following is represented by frogs? Frogs are advanced life forms, or at least they're animals. Certainly they're not bacteria or fungi or anything like that. And they represent the diploid life cycle, which is mitosis, in the idea that there are two pairs of chromosomes, or pairs of chromosomes, for every chromosome that they have. And so therefore the correct answer is B. Which of the following plays a role in cancer development? Not controlled cell division, which is okay, but mutations is certainly the correct answer. CDKs do not stimulate cell division. Wearing sunscreen is actually good for you. That's certainly not going to cause cancer. The problem that does cause cancer could be mutations, so that's B as well.